Okay, so let's take a look at 1532 now. Mutations in the I or 1L2R gene, or IL, let's see. I want to change back to my yellow pen, sorry everyone. So IL2RG gene in humans, and genes in humans seem always have these long names that are kind of meaningless, right? IL2RG. So maybe this is like interleukin or something. Anyways, we're dealing with a gene involved in immunity in humans. So, okay, so mutations in this gene cause approximately 30% of severe combined immunodeficiency disorder. So this is where your immune system does not work at all, almost. And I believe this is where you have to be isolated from external environments and all sorts of germs, and you pretty much your immune system does not work. I think we need to look this up on Wikipedia to be sure, but this might be the bubble disease, where you have to live in a bubble, protected from any germs in the environment. Anyways, 30% of SID cases, or SCID, I don't know if it's my SCID, I'll call that. SID is sudden infant death syndrome, or SIDS, and this is SCID, I guess. I'll call it SCID. So 30% of SCID cases. So these mutations result in alterations to a protein component of the cytokine receptors that are essential for proper development of the immune system. So you have a mutation here, your immune system does not properly develop. So the IL2RG gene is composed of 8 exons and contains upstream and downstream sequences that are necessary for proper transcription and translation. Okay, so here's our gene here. I'll diagram this. Okay, we have our +1 site where transcription starts. We'll have our transcription termination site. Now we're going to have 8 exons. What did it say? 8 exons. Let's see. I forgot already. 8 exons. Okay, so that means 7 introns. Okay, let's put an intron. Let's put an exon here. 1, 2, and in humans the introns are usually really long, longer than the exons. Actually, I say usually, but I don't know that for sure. They are sometimes long, and I imagine they must sometimes be short too. So 1, 2, 3, 4. I'm running out of space here, aren't I? 5, 6. Okay, I'm going to make the gene a little longer here. 7 and exon 8. Okay, so this is our hypothetical sketch of the IL2RG gene. The promoter would be up here, +1 here, and here's all the approximate locations of exon sequences and transcription termination sequences here. Okay, so now let's take a look. Below are some mutations observed. Okay, I think I can paste this in here. Yep, here we go. Okay, we don't need to see that. So here are some mutations. Now what we need to do is one second here. Below are some of the mutations observed. For each, explain its likely influence on the IL2RG gene product, that means protein. Assume its length to be 375 amino acids. Okay. So, this gene right here makes a transcript that is, it makes a transcript here's our transcript with a 7-methylguanosine cap and poly A tail, and there's going to be a start codon and a stop codon. I'm guessing it's UAG. And this coding region here, coding sequence, codes for a protein that is 375 amino acids long. So we can multiply that by 3 and have an idea of how long the coding region is. So 375 times 3, it's going to be 15, 22, 11. So 1,125 nucleotides long. 1125. Plus the UAG, the stop codon, so 1128 nucleotides. Okay, so I think we're ready to take a look at these mutations. So explain its likely influence on the IL-2R gene product. Okay, so a nonsense mutation in the coding regions. Okay, so what would that do? Okay, that seems pretty... It doesn't seem like that much information. Let me take another look at the question. Okay, so all of these mutations lead to severe combined immunodeficiency syndrome. Is this severe? Yep, severe combined immunodeficiency disorder. So a nonsense mutation in the coding regions. Okay, pretty easy, right? So why would a nonsense mutation in the coding regions, what would that do to the protein? Well, nonsense mutation, okay, let's say nonsense mutation is going from a codon that codes for an amino acid to a codon that codes for a stop. Okay, so let's say we get a stop here, okay, well then let's say that translates to this spot right here, then that would lead to a truncated protein that couldn't function. So any nonsense mutation is going to decrease the length of the protein, and maybe these amino acids that are coded down here are extremely important to the function of the protein. They've been conserved, so they probably are important. So any nonsense mutation is going to truncate the protein, which means makes it shorter, and it's not going to function. So that's probably how a nonsense mutation influences the gene and the cause of this disorder. So an insertion in exon 1 causing a frame shift. Okay, so here's exon 1. Let's say the start codon is right here. Okay, so an insertion in exon 1, let's say insert somewhere down here. So what is that going to do if it causes a frame shift? It's going to change the codons for everything. If this little piece right here, if the frame shift is happening right here, it's going to change the reading frame for every codon, and every codon downstream of that frame shift is going to now code for probably code for a different amino acid, right? So if we had CAGs in a row and we insert some bases, say we insert an A right here. Okay, so now we have CAG, ACA, GCA, GCAG. So here, before we inserted that A, we had a reading frame of CAG. This might code for glutamine, I'm not sure. We need to check that to know for sure. So we had a frame shift, inserted an A, and now we have, okay, this one codes the same, but oh, this one's different. We have to look up, and this one's different, and this one's different. Oh, they're all different. Okay, so a frame shift very early in the protein is going to change all the amino acids, and it's not going to be functional, not likely to be functional. So an insertion in exon 7 causing a frame shift. So exon 7, this is 1, 2, 3, 4, 5, 6, 7. Okay, an insertion in here, it would seem like it would be less likely to have an effect on the protein than an insertion in exon 1. Causing a frame shift here is only going to change the amino acids that are downstream of exon 7. So one thing I'm confused about with this question is, are these real mutations? Are we supposed to decide which ones are more likely to have an effect and cause the disorder, and which ones are less likely to have an effect? Now, apparently they all have an effect. Okay, so we're trying to figure out why all of these have an effect. So it was easy to see with 1. With 7, okay, so we're going to change, all of the amino acids. If a frame shift, something inserts in exon 7 and it causes a frame shift, well, we're changing all the amino acids that are coded by bases downstream of that insertion. And apparently that has an effect because, with respect to this protein, the amino acids at the end of this protein are important for its function. So a missense mutation, what would that do? So a missense mutation anywhere is changing an amino acid from a codon that codes for, let's say, alanine to a codon that codes for any of the other 20 amino acids. So alanine to leucine. Or maybe the codon originally codes for phenylalanine, and now we're coding for tyrosine. So that's a missense mutation, and so a missense mutation can, by changing an amino acid in the protein, well, each amino acid, could be important for the function of that protein, and some of them are going to be critically important. So it makes sense changing one of the codons to causing a missense mutation in one of the codons will change the amino acid, and that could have an effect on the function of the protein. Deletion in exon 2 causing a frame shift. Okay, well that's pretty easy, right? So you delete 1 or 2 bases or a certain number that changes the reading frame. You cause every codon downstream of that mutation to code for a different amino acid, and it's pretty easy to see how that would inhibit the normal function of the protein. So what about deletion in exon 2 in frame? Well, that could also influence the function of the protein if, those amino acids happen to be important for the way the protein functions, the way the protein folds. Sometimes you can delete a couple amino acids from a protein, and as long as it doesn't cause a frame shift, that protein can still function, still fold properly. Maybe it won't function as well as wild type, maybe it will, maybe it functions slightly, just it's not completely like wild type, but we don't have assays that can quite tell that it's not functioning as a wild type protein. But in some cases you can delete things that are important too, and you can completely lose the function of the protein, even if you're just deleting a couple of amino acids. So even when you're not causing a frame shift, deletions or insertions can disrupt the overall folding, or can delete amino acids that are critically important for the function of that protein. And so a large deletion covering exons 2 and 3. So with that one, what could happen there? So again, that's just deleting a larger portion of the protein, so we're getting rid of a lot of amino acids, and of course the bigger the deletion, the more likely you are to interfere with the function of the protein. So I'm not a big fan of this question. I guess it allows us to look at the different types of mutations that can occur to a gene, and discuss or think about how they could influence the function of a protein, and they can all have negative effects depending on where they occur and how important those particular amino acids were to the function of the protein. And now geneticists are finding now even there are some things about transcription and transcription rates where even before we get to translation where certain bases that are in the gene or being transcribed can interfere with transcription rates and translation rates and things like that. So there's a whole another advanced level that we're not going to discuss in this course, because this stuff is really new and scientists are just learning about it now. But all this stuff we just talked about here, this is all true. And these are all different ways mutations can influence the function of a protein.